And so we're going to touch on more of the specifics of how to tailor these different applications later on this afternoon. But this morning we're going to focus mostly on using these databases for optimizing a basic qPCR reaction. So just a general overview of our SciTools. This is available on our IDT website. We have several tools available here that are able to assist with qPCR design. We have the real-time PCR tool, which real-time PCR tool, which is good if you're just designing a basic assay. You just want to stick in your gene of interest and have it pop out an assay to use. Our PrimerQuest tool, which we'll be focusing on today, is a tool which allows for a lot more optimization and customization. So if you want to target your primers and probes to specific regions, if you have a particular sequence context you need to design around, the PrimerQuest tool is good because it allows for a lot of customization. And then we have our OligoAnalyzer tool, which is good for analyzing your primers and probes after you have your design. You can go back and check your primers and probes to make sure that there's no secondary structure, such as hairpins and dimers and heterodimers among your different reactions that can interfere with your qPCR reaction. So we're going to go through some of these tools today in this session. So broad overview of the NCBI databases. The NCBI databases can be very useful for designing these qPCR assays. You can go to the NCBI database to download sequence information for your gene of interest through either the NCBI nucleotide or gene databases. You can also use NCBI to perform the BLAST search to look for assay specificity, and finally search for presence of SNPs in your probes, and all of this can be done right within NCBI. So in this first segment, we're going to talk about using the NCBI databases for custom qPCR design, and with the focus of getting the sequence information for your gene of interest in a highly usable form. So I'm sure many of you are familiar with NCBI. If nothing else, you've probably gone to download some papers from PubMed or searched around a little bit for your sequences. So some of this may be a review for you, but we're going to kind of just broadly go over what kind of information is accessible in NCBI and how to access that information. So the NCBI, or National Center for Biotechnology and Information, was founded in 1988 as part of the United States National Library of Medicine. And it houses a series of databases that are relevant to biotechnology and biomedicine. These include GenBank, which is a database of over 1 × 10⁻12 base pairs of DNA sequences. The Gene Database, which then integrates that information and provides gene-specific information from numerous different species. Also database SNP, which is a database of all of the reported single nucleotide polymorphisms, or SNPs. NCBI also contains the BLAST sequence similarity search program, and maintains PubMed, a journal database for biomedical literature, and much, much more. These are just some of the different databases that we're going to look at today. So to begin, we can go to NCBI. This is the NCBI homepage. We can search for our gene of interest. You can either input your gene of interest by name here, or by accession number if you know that. And you can search by either the gene database or the nucleotide database. And so we'll look at some of the different outputs from these shortly. And then you can see also on the NCBI landing page easy access to all these other databases that we will be talking about in terms of qPCR design. Your BLAST search, checking for SNPs, and so on. So the NCBI sequence files can be entered by anyone. Because of this, they may or may not be checked for accuracy. So always use caution when you're pulling your sequences out of NCBI. They may contain contaminated sequence, whether it be plasmid or otherwise, and may contain annotation errors. All of the sequences in NCBI are denoted with unique identifiers or accession numbers. These accession numbers are the letters that are at the beginning of the file for the particular sequence, and they indicate the type of file that you're looking at. So nucleotide sequences, which we'll be primarily concerned with today, start with one or two letters, such as is shown right here for this human CD81 mRNA. It starts with an NM, and then it's a unique identifier number. The RefSeq database is a good place to get your sequence information. It is non-redundant, explicitly links nucleotide and protein sequences, and these sequences have ongoing curation by the NCBI staff and collaborators with reviewed records. And these different data validations are tracked and ensured accuracy and format consistency. And so these have distinct accession numbers. All of them will include an underscore character. And then, again, you can access the different versions that have been tracked, so you can go back and look at previous accession numbers for these same genes. Yeah.
So you mentioned annotation errors in the previous slide. So with respect to a reference sequence, what is an example of an annotation error?
So in this case, in the RefSeq database, that's much less of a concern. These
Okay.
are curated, annotated, and tracked. It's more, and actually I think the next slide, yep. So any of the sequences that have like an NM accession number have been curated, so they've been checked for accuracy. It's more of sequences like the AF or direct submissions, which have a different accession number, like
Or ESTs?
ESTs, yep. So anybody can just download and dump their sequence information into NCBI. So if you're looking at if you do a blast search and you come up with a sequence that has an identifier of AF or something that's not one of your standard identifiers, then you might have to worry about the accuracy of that sequence. But if you're looking at an NM, a curated mRNA sequence, it's the best place to go to get your sequence of interest because it has been curated and annotated. Okay. So here's just a list of some of those different accession numbers, as I mentioned. Anything that begins with an N, so NM, NP, and NR, has been curated. The M is for the mRNA sequences. Proteins will have an NP, and then the R's will be for the curated non-coding RNAs. Anything you see with an X in front of it is a predicted mRNA, a predicted protein, or a predicted non-coding sequence. Then the genomic sequences are denoted with the NG accession. Chromosome, so microbial replicons, organelle genomes, human chromosomes will have the NC designation, followed by their unique number. And then alternate assemblies will have an AC. And then we have the big contigs and big assemblies, which have NT or NW. So in most cases, when you're designing your qPCR assays, you'll want to look for these NM sequences in the database. So how do we find this information in NCBI? Again, if you go to the NCBI website, you can search by either gene or nucleotide. This will take you to two different landing pages, both of which you can access the accession file for your gene of interest. You can search by the unique accession number or simply by your organism and gene of interest. So here's an example of the information that we'd see from searching for mouse gap DH by gene. And so it takes us to this page where we can see on chromosome 6 and the other genes that are surrounding that. And then we get a nice graphic representation of the gene organization, so the intron and exon arrangements for our gene of interest here. And then on this page, if you scroll down at the bottom, it has the unique accession numbers, the NM numbers that you can click on and directly get your sequence from there. Alternatively, we can search for our sequence of interest in the nucleotide database. And so these files will bring up mRNA and genomic sequences as well as transcript variants. So you want to look for an NM of your gene of interest and pull up that sequence. And what it will provide for you is your sequence of interest. Here's the unique accession number. It tells you a little bit about this gene and some of the references associated with it. But then you can also obtain the FASTA sequence in an easy, accessible form that's easy to put into other programs and manipulate it for other applications. We can also go to see a graphic representation, again, of our gene organization with the introns and exons. And then here's that FASTA sequence. So this FASTA sequence is highly versatile. It's easy to just copy and paste and put into PrimerQuest, for example. And so that's just a general overview of how to access our sequence of interest. We're bringing it over to PrimerQuest. So we're going to focus now on PrimerQuest and the different things that we can do within PrimerQuest to generate a customized qPCR assay. So the first thing that you need to do in going to PrimerQuest is to input your sequence of interest. So you can either input the FASTA form that we were just talking about, or you can put in the accession number. So you can download either by sequence file, or you can click here and enter your sequence identifier by the accession number, and then it will pull the data from NCBI and upload your sequence of interest into PrimerQuest. So once you have your sequence uploaded, 3 default buttons will appear, either for PCR, qPCR, and then the qPCR intercalating dyes. Today we're going to focus on designing qPCR assays which are probe-based, so we're going to select our qPCR primers and probes option. So if you just want a basic qPCR assay, you don't need to target it to any particular location. No specific changes are necessary. You can just click this orange button here, and it will generate a list of potential qPCR assays for you. For each of these assays, then, you can click on View Assay Details, and it will show you the sequences of the primers and the probes for each of these assays. You can see on the top, the output shows you the approximate location for each of these assays, so you can get an idea of where within your sequence the assay is located. There's also some potential for customizing these based on specific needs. If you click on the Custom Design Parameters option, I would first warn that you make sure that the qPCR button is clicked here before changing any of the other parameters, because after you've changed some parameters, if you realize that the right option is not selected and you click a different one, it goes back to the defaults, so you'd have to change everything else back again. So first thing we do is select our qPCR option if it's not already changed. Here you can increase the number of results that are returned. It defaults to 5. Often it's useful just to increase this to anywhere up to 50, but even 10 or 15 reactions, so that you have some reactions to look through and more choice in where those are located. You can also vary the primer locations, how variable the primer locations are in the output. So either the forward or reverse primer, if you turn this to up to high, then they're going to be spread throughout your sequence more, or low, they're going to be targeted more specifically. And so then you can also change the specifics for your reaction conditions. These defaults to divalent salt of 3 millimolar, DNTPs of 0.8. This is kind of standard for a lot of the qPCR master mixes out there. If you know for a fact that yours differs from that, you can go in and make those changes there. And so at the bottom part of our PrimerQuest in the customized options, there's a region where you can target the location of your assay. So you can tell it where you want your probe to lay down, or primers to lay down, or excluded regions where you want to avoid. So here, if we want to tell our probe, and we'll go through more specific examples of how to do this later this afternoon. But if you want your probe to lie down within a particular region, you can exclude the rest of your sequence from that to kind of target where the assay is going to designate where your probe is laying down. If you want to exclude an entire region, you can do that here, and that will exclude primers or probes from laying down in that region. An included region will force the assay to be placed in that area. And then you can also design across junctions. So if you want to design across an exon-exon boundary, you can put those boundaries in here to make sure that the assays are designed across those junctions. And so here's an example where we're tailoring the assay to different regions. So here's the basic output when we just do the qPCR basic settings. Now if we set our excluded region to 260 to 280, you see that it's not putting any of the primers in that region, but our probe is still located in that region. However, if we exclude the probe from 260 to 280, our excluded region of the probe, now it moves the probe and the assay out of that region completely. Alternatively, you can target that region 260 to 280, and all of your assays, that output will be targeted to that area. So if your target is particularly biased in terms of sequence, if it has a lot of AT or GC content, you may need to change the primers. You may need to change your primer or probe parameters. For example, if you have a highly AT-rich sequence, you may need to increase the length of your primers beyond the 30 nucleotide default. You can do that here. If you have a really high percent GC or a really low percent GC that you're not getting any assays because of the sequence context, you can come and change that here. These are kind of the standard defaults that are set to optimize the qPCR reaction, but you can change these as needed based on your sequence that you're working with. Also down here, you can see it defaults the amplicon size between 75 and 150 bases, and that can be tailored too. That's just the general default recommendation. So once we have our original design, we've got our output list from PrimerQuest, and we can select through those assays. We recommend starting with the first assay. PrimerQuest will kind of rank them based on score, and the highest scoring assay will always be on the top. So we recommend then checking to make sure that that assay is specific by doing a blast search, and then also ensuring that there are no SNPs to worry about within the primer and probe sequences by looking in database SNP. And then after you've ensured that that assay is highly specific, you can check the primers and probes for secondary structure and dimer formation. PrimerQuest already does check for this, and that's based, or a factor that's considered in the scoring. However, it scores on a lot of different things, and so an assay which may have a minor hairpin or dimer issue might score higher than another assay just based on all of the different factors that it takes into account. So it's always recommended that if your goal is to design a highly robust and most optimized qPCR assay, that you do go back and check the primers and probe for secondary structure and dimer formation using OligoAnalyzer. So we'll go through using NCBI BLAST to check for primer specificity. So again, if we go to the NCBI homepage, you can access BLAST right from the homepage here, or you can type in the unique address for BLAST. So what is BLAST? BLAST stands for Basic Local Alignment Search Tool. And again, it's found on the NCBI database. BLAST will align a user-defined query or sequence to a wide variety of different databases and can translate the query or the database to align the sequences. You can put in one sequence or two or more sequences for BLAST to align. And it runs on a heuristic algorithm that creates alignments very quickly by breaking the sequences into words and then searching the database for matches to those words. It chunks up your sequence of interest into smaller portions. And then reassemble these back based on the matches and the criteria that's entered. So from the BLAST site, you can see that you can search a specific species of interest if your species of interest is there. Otherwise, you can do a generic nucleotide BLAST. And so again, BLAST divides the query into subsets called words, which the algorithm then uses to perform the alignment. Here's an example of a 35 nucleotide sequence. You can set BLAST to look for different number of words. So 7 letters is one of the settings and the one that we recommend using when you're blasting. So here's an example where we have a 7-letter word. And what BLAST does is break this sequence up into all of the possible 7-letter words that it can from this sequence by moving that red box over one base at a time. And so in this 35 nucleotide sequence, the max number of words possible is 29 different words. So all of these possible words that can be generated from the sequence are then used by the BLAST algorithm to create the alignment. And so in the BLAST output, it has multiple different hits or anything that matches your sequence of interest. It's any sequence in which the query is aligned and is returned in your BLAST results. Then the identity is the extent of exact matches between these two sequences. For example, ACGT and ACGG have 75% identity. So each of the different hits will get a score by BLAST. So similarity indicates positives in BLAST scoring. And then these scores, the BLAST gives it a raw score, and that's converted to a bit score based on the statistics described in Carlin and Altschul, which if you're into those kind of statistics and want to know exactly how BLAST is calculating all of these things, which I'm not a mathematician, but those are available in that publication there. I just like to look at my BLAST results and get an idea for what's most similar or least similar to my sequence of interest. A high score does not always indicate that the query is unique, however, so it's important to really look at the data that BLAST gives you and keep in mind that just because it gives a high score, it doesn't indicate a unique sequence. The score is only dependent on the alignment, the length of sequence, and the length of the database. So BLAST will give an e-value, which is the expected amount of random sequences that have equivalent sequence alignment. And this is calculated using the max bit score combined with the length of the query and the database. And it tells you the relative strength of the alignment. So when you're looking at your BLAST outputs, you can scroll through them to see the breaks in these changes in this e-value. Shorter sequences will always have a higher e-value because the probability of finding that sequence is going to be much higher. And a low e-value does not mean that you have a unique match necessarily. So when inputting our primers and probes into BLAST to check for specificity, we don't want to do just one primer at a time because it might one individual primer might hit a bunch of different things that it is very similar to, but in terms of a qPCR assay, that isn't what we're concerned about. We want to know what amplicons, what other non-specific amplicons can be generated by our specific primer combination. And so the best way to do this is to input your primer sequences into BLAST, you're separating your forward and reverse primer by several ends. Such as this. And so we can see here we've got our forward primer, our reverse primer, separated by a series of ends. We can choose the data. Oh, sorry.
Does the number of ends have to be specific to the amplicon size?
No, just variable. Yeah, you just want to make sure it's greater than your word, whatever word size that you have selected. So then from here we can choose which database we're going to BLAST. If you know you're looking at a human or mouse specific sequence, you can select that. Otherwise, you can select the nucleotide others option, and then you can target an organism here under organism if you wish. If you don't specify the organism, you're going to get a lot more output to scroll through. Yes?
So your reverse primer sequence should be reversed when you put that in there?
It should be, yeah, it can be. Otherwise, just keep in mind that instead of having a plus plus and a plus minus output, they're both going to be plus plus. But ideally, yes, you'll put the forward primer forward and the reverse primer in the reverse orientation.
So the reverse primer, you don't reverse complement it. That's what you're saying, or you actually?
Yes. Yes, you can. You'll get the same BLAST output whether or not you keep the primer in reverse complementation or not, but your output will look different. So it's best to reverse complement your primer. So the PrimerQuest assay will spit out a forward primer and a reverse primer.
Already.
In the assay, it's already reverse complement.
So that is what you use
and that's
then
what you
in the
put in
BLAST?
there. Yes.
Okay.
Because I think BLAST is searching on both strands and it's kind of keeping it in conjunction with the forward primer. So kind of then keeps that moving as you do. So yeah. It just saves a step of having to BLAST all the time, kind of doing it.
So if you know your species has a completed genome project, you can consider using this NCBI genomes to limit the BLAST results as well. So any categories here that you can select will limit your overall output and just have an easier amount of data for you to look through. So we can change the parameters a little bit for the BLAST scoring. We want to select the less rigorous algorithm, so the somewhat similar sequences or BLAST-N algorithm, and then change that word size to 7 so that it's chunking up the sequence in smaller regions. And then here's an example of what the BLAST output will look like. Each of these different lines indicates a unique match to your forward and reverse primer. You can scroll over these in NCBI and it will tell you which, the name of the sequence that it is hitting. So the graphic summary can give you an overall sense of what the results are, how many hits, how many unique hits there are to your primer and primer combination. And then we can get some more detailed information about each of these by scrolling down in BLAST. And look at the results list. So here's the list of what each of those different lines represents. you can see by looking at the top, the transcripts that our primers are hitting. The primers seem to be specific to our target of interest, which is HMGA1. It's showing the various different transcript variants for HMGA1. And then we can look at the genomic sequences to make sure that it's only hitting a single chromosome. If our assay is going to be highly specific, it should hit one chromosome. You can see here we're hitting several different chromosomes. So we need to look at each of these to see what might be going on. You can ignore the alternate chromosome assemblies and just focus on these primary assemblies here. So you can see here's a hit on primary assembly chromosome 6. And there's one for chromosome 4 and chromosome 2 as well. And so by looking at the overall e-value and query coverage and looking for jumps in either of these, you can get a sense of what other sequences might be a potential target for your primer and your primers. By opening up the individual hits and looking at the graphics window, you can see where each of your primers is actually laying down on these different sequences. You want to make sure to check the distance between the primers binding to make sure that it's an actual potential threat. So if there's a huge difference between where the forward and reverse primer lays down and an amplicon isn't ever going to actually form there, then it's not something that you need to worry about. And so here you can see by clicking on that graphics view, Blast will open a window. And it's nice because it lays down our sequence right here that we input alongside of the transcript. And so you can zoom in or out here using that. You can zoom out with the negative sign. You can zoom into the sequence level, which we're at right now with the plus sign. And you can grab within this window and drag the sequence side by side. Also convenient here is that you can configure here for SNPs, which I will show in just a little bit as well. So you can actually do your blast alignment and checking for potential SNPs all at one time. And so you can see here our assay. If we zoom out, you can see our primers are denoted by those little gray boxes with the black arrows here and on the other side, if I can get my mouse to work. And I can't, so on the other side as well. And then we can see that each of these is laying down in a different exon, so that looks very promising. Our assay is spanning 2 different exons, which is great. Here is one of the other hits on chromosome 2. However, it looks like our primers are aligning to a pseudogene that doesn't look transcribed. The real transcripts are going to be denoted in this green color. Non-transcribed transcripts are going to be in this gray color. So our primers are aligning to a pseudogene, and the red bars will indicate a mismatch. And then, again, here's another pseudogene. But then up here, you can see the primers are also aligning to this, which looks like the green line, which looks like a real transcript. So what is that? So if we look at that more closely, you can see that it is a real transcript. And that those primers are aligning in an intron of what is another real transcript. So that's something that we want to be careful of and redesign or avoid if possible, because it has the potential to be, has the potential to be in your prep. And so as I briefly mentioned, you can also use NCBI here to check for the presence of SNPs in your primers. And you can do this from this blast page by configuring, using a configure button, and that will pop up this window here. You can select on variation, and that will bring up a window where you can check this box next to SNP. And once we do that, all of the SNPs will appear in this graphic representation. So you can look and see if there's any SNPs that overlap with your primer sequences. So it looks like here we have one potential SNP. So by clicking on that identifier, it pops up this window. And then this right here is a link to that SNP page. So we can click on that to get more information about this potential SNP. And so from this page, you can see that SNP is a single base substitution. The ancestral allele is a T, the mutant allele is a C. There's no allelic frequency reported for this SNP. And then if you scroll down to the bottom of the page, you can see sometimes more specific frequency data. So by looking at these, we can kind of roughly divide these SNPs in our primers by potential risk. So just because you see a SNP in your primer doesn't mean you have to automatically avoid that primer. You want to look at the SNPs, look at their allelic frequency, and kind of make an assessment as to whether or not you think that SNP is likely to be in your population and is likely to be problematic for you. So here's an example of a reported SNP with a allelic frequency of 0. So if I see this in my assay, I probably wouldn't worry too much about it. In fact, I would just keep the assay as is. It's also data from 1,000 genomes, which is a highly trusted source. And here's another one, which has no allelic frequency data. These you kind of have to be a little more cautious about. Somebody has reported this SNP exists, but we don't have any allelic frequency data. If you can't scroll down to the bottom and get any more information, these are kind of proceed with caution. Usually they're okay, but if you do notice some problems with your assay, it's important to keep these steps in mind. But overall, the ones that don't have any allelic frequency or other data or information with them are not likely going to be problematic. Then here's one that's reported at 0.244 allelic frequency. This is something you would want to avoid. It poses a significant risk, and look to redesign it if possible. So then I would probably go back to PrimerQuest, select the second assay on the list, and repeat the blasting and SNP checking with that second assay. And so you continue that process until you find one that is highly specific by blast and has no SNPs with a significant allelic frequency in either your primers or probes. And I did forget to mention, when you input your primers into BLAST, you can also put your probe in there as well. So you can put your forward primer, several ends, your probe, several ends, and your reverse primer, and blast them all together and check for SNPs for all of them at the same time. And so now we're going to go through how to use OligoAnalyzer to check these primers and probes. So we've got a good assay. It's highly specific. Oh, does anybody have any questions on any of the NCBI stuff before I go back into PrimerQuest and OligoAnalyzer? Okay. And so we've got our assay. We know it's highly specific by blast. We don't have any SNPs in our primers or probes that could potentially interfere. So now the last thing that we want to do is to check these primers and probes using the OligoAnalyzer tool to assess whether there are any dimers or hairpins that might potentially inhibit the reaction. And so the PrimerQuest designs that come out are always going to give you the best assays for that region that you specified. So it's going to use the sequence that you give it, though. So if you're giving it a difficult sequence context to work with, the assays that come out may not always be the perfect assays, but they're the best assays for that sequence that you are giving PrimerQuest. And so PrimerQuest itself will check for self and heterodimers, But again, this is only a small part of the scoring system that's used. It also scores based on amplicon length and TM and GC content and all of those things. And based on all of those different scoring parameters, PrimerQuest will put the top scoring assay first. So that doesn't always mean that you can guarantee that that assay won't have some small secondary structure or dimer issues. So it's always best just to make sure and go back and check these things. So we go to the OligoAnalyzer tool, and from there, input our primer and probe sequences to perform self-dimer checks for the primers and the probe, heterodimer checks on all of the primer and probe combinations. This is especially important if you're going to be multiplexing. So you want to check, if you're multiplexing multiple primers and probes in one tube, you want to make sure to do this heterodimer check and for all of the different combinations of all of the different primers and probes. And then also check for hairpin structures. Our general cutoff is anything with a stability less than 9 kilocalories per mole, or multiple hairpins forming with a stability of less than 4 kilocalories per mole. And so here's an example of some dimer output from OligoAnalyzer. So this is specifically self-dimer. So you can see here, here's a delta G value of minus 15.42. That's lower than our minus 9 cutoff. So this would be something that we look at and look at more carefully. It looks stable, again, but it's not overly dangerous. You can avoid it if possible, but if this is the only assay that you have to work with, I wouldn't throw that assay out altogether based on this dimer. Here's another example where it looks stable at minus 9 kcal per mole, but it looks, by number, looks less dangerous than this one. However, despite this higher number, it's not a problem because it's not extendable. So this won't be able to extend. So even if this does form, it's not going to extend and pose a problem for your assay. It's important to keep in mind that that minus 9 cutoff indicates 1% of your primer combination. So if at this minus 9, 1% of it is going to be tied up. And here's one at minus 6. It doesn't look stable. However, this right here is the most dangerous possibility. Has a danger of exponential amplification. So even though that number is only negative 6.3, this is something that I would worry about and most likely redesign the assay to avoid. Because even 1% or less than 1% of this, when amplified in your reaction, could be very problematic.
So just to let you know too, IDT, whatever you send us, we're going to try to make. You may not know that it's a really difficult synthesis to make until our third attempt, at which time you're like, where is this thing? Okay? So if you can do your homework ahead of time and find something that doesn't have primer, then that's going to help us get you code a lot quicker and something that's going to work a lot better in your experiment. So this is really important stuff.
All right, so that's assessing dimers. And then moving on to looking at secondary structure. The hairpin function in OligoAnalyzer pulls from unifold predictions. So it makes predictions on potential secondary structure or hairpins using unifold. And so a window will pop up here, which shows all the possible secondary structure for your sequence of interest, for your primer or probe. And then you can use this information to kind of judge the risk of how much of your primer or probe is going to be tied up in this secondary structure. So here's an example here where it's got a relatively high TM. We kind of put a loose cutoff at 40 degrees. Anything over 40 degrees we want to look at more carefully and consider whether or not that's going to be a problem for our reaction. However, here, that TM can often be an overestimate when you have just a few bases that are matching. So here we only have a stem of 4 bases. So this is most likely an overestimate. The delta G is still relatively well below that -9 cutoff. So this is not likely to be a major problem for the assay. But if you were to see a hairpin structure with a very high TM and that delta G approaching -9, that's something where you should be concerned and perhaps look at other primer or probe options. Go to the next assay in PrimerQuest and look at those. And so finally, that was kind of some information if you need to or want to design assays yourself using the tools available between the NCBI database and our IDT PrimerQuest. However, if you just need a basic qPCR assay for your gene of interest and you're looking in human, mouse, or rat, we do have a database of a lot of already designed assays for you. So you can go to the front-end qPCR database and put your gene symbol or accession number and organism of interest and come up with a good assay for your target gene. And so as Rita already mentioned what the parameters were that those assays, primers and probes, are based off of. And so the output will come up and you have several different assays to pick from right there for whatever your gene of interest might be.